Hello all, a very warm welcome to another video. In this session, we will discuss the fourth part of light reaction. In the previous parts, I have explained the site and machinery of light reaction, photolysis of water, electron transport and formation of NADPH. If you wish to watch those videos, you can get the link from the description. Let's move to the part 4. In this part 4, we will mainly focus the first event of ATP synthesis, that is proton gradient. Let's directly move to the topic. As you know, the process of light reaction occurs in the thylakoid. You know, this is thylakoid membrane and this side is thylakoid lumen. On this thylakoid membrane, we have seen two photosystems, PS1 and PS2. In between these two photosystem, there is another multi-protein complex, that is cytochrome. During the process of light reaction, we have seen photolysis of water or splitting of water at the luminal side of PS2. That means water molecule is split into 2H++ half O2. And the removed electron are transported through an electron transport system. You know, there is an electron transport system. The removed electrons are transported through an electron transport system to NADP+. That finally resulted in the formation of an NADPH. You know, this is a first product of light reaction, that is NADPH. Then how to synthesize ATP? ATP is also another product of light reaction. Now we have to check the synthesis of ATP. Formation of ATP requires an enzyme. That enzyme is called ATP synthase. This ATP synthase is also located on thylakoid membrane. This is called ATP synthase. And this ATP synthase is having mainly two portions, FO portion and F1 portion. Then how to synthesize ATP using this ATP synthase? What is the driving force behind the synthesis of ATP? Remember, the driving force behind the synthesis of ATP is nothing but a proton gradient. Then what is proton gradient and how this proton gradient is created? Proton gradient is nothing but the difference in concentration of H+ ions across the thylakoid membrane. You know, this is the thylakoid membrane. Internal to thylakoid membrane, we have thylakoid lumen. And outside thylakoid membrane, there is another space, that is stroma. Proton gradient is nothing but the difference in concentration of H+ ions across the thylakoid membrane. That means concentration of H+ ion should be more in thylakoid lumen, concentration of H+ ion should be less in the stroma. This difference in concentration of H+ ion across the thylakoid membrane is called proton gradient. Then how to create this proton gradient? Formation of this proton gradient depends on two oxidations and two reduction. First, we will check what are all the two oxidations which can contribute to the proton gradient. The first one is oxidation of water or photolysis of water. As you know, H2O is split into 2H++ half O2 plus 2 electron. You know, this photolysis of water occurs in thylakoid lumen. Since this process occurs in thylakoid lumen, within the thylakoid, concentration of H+ ion increases. You know, this is photolysis of water. During this process, 2H++ half O2 will be released. And this process occurs inside the thylakoid lumen. As a result, concentration of H+ ions increases inside thylakoid lumen. The second oxidation is oxidation of PQH2. Then what is the result of this oxidation of PQH2? PQH2 is oxidized to PQ plus 2H++, 2 electron. Then where can you see these 2H+ ion? These two H+ ions also transported to thylakoid lumen. The third part of light reaction, you have seen PQH2. This PQH2 is a mobile electron carrier. Once this PQH2 is transferred to cytochrome, Within the cytochrome, PQH2 undergo oxidation. As a result of oxidation of PQH2, this PQH2 releases H plus ion. This H plus ion is also transferred to thylakoid lumen. You know, this is thylakoid lumen. Within the thylakoid lumen, concentration of H plus ion increases. So oxidation of PQH2 also increases the concentration of H plus ions within the lumen. These two oxidations increase the concentration of H plus ions within the thylakoid lumen. What are the two reductions which will contribute to the proton gradient? Let's check. Two reductions. The first reduction is reduction of PQ. During reduction of PQ, PQ accepts 2 H plus ion plus 2 electron and is reduced to PQH2. This we have explained in the part 2 of light reaction. You know, during reduction of PQ accepts 2 H plus ions from stroma. As it receives H plus ions from stroma, concentration of H plus ions within the stroma decreases. You know, PQ is present in the PS2. After accepting 2 electron, this PQ accepts 2 H plus ions from the stroma. This part I have already explained in the second part. I think you remember this. If you didn't watch, please watch the second part of light reaction. During this reduction of PQ, H plus ions are accepted from stroma. So concentration of H plus ions in the stroma decreases. That is one of the reasons to reduce the concentration of H plus ions within the stroma. The second reduction is nothing but reduction of NADP plus. Reduction of NADP plus. During reduction of NADP plus accept H plus ions plus 2 electron and is reduced to NADPH. I think you remember the formation of NADPH that we have explained in the part 3 of light reaction. During the formation of NADPH, NADP plus accepts H plus ion and 2 electron to give rise to NADPH. You know, this H plus ions also comes from stroma. This is also coming from stroma. So what happens in the stroma? Within the stroma, H plus ion concentration decreases because H plus is used for producing NADPH. So we can conclude these two reductions reduce the H plus ion concentration within the stroma. As a result of these two oxidations and reduction, a proton gradient is created across the thylakoid membrane. That means concentration of H plus ions increases in the thylakoid lumen and concentration of H plus ions decreases in the stroma. This difference in concentration of H plus ion across the thylakoid membrane is called proton gradient. How to synthesize ATP using this proton gradient? That we will discuss in the part 5 of light reaction. You can get the link from the description. If you like the video, please subscribe and enable the bell icon to get my upcoming videos on NEET and CSIR UGC NET exam topics.